Welcome everyone to the second podcast on postdocs of Washington University in St. Louis. Today we will interview Dr. Fernanda. Details to her publications and webpage are in the description of this video. So let's talk to her. Welcome Fernanda to our podcast. It's a pleasure to have you here today. Could you please tell us about your professional and educational background?
So thank you for inviting me. So I'm originally from Brazil. So I got my bachelor's degree in biotechnology there at Federal University of Pelotas, which is where I'm from, my hometown. And then back then I worked with multiple things, undergrads trying to get as much experience as they can. And I was part of an oncology group there. We did a lot of mostly wet lab research. Then from there, I got a scholarship to come study in the US in 2012. It was like my last year of my undergrad. And I worked with a pig model for cancer, which is very different than what I do for now. A lot of wet lab and cloning and transgenic work for this inducible model for cancer research. And then that opened the doors for me for staying over here. So I finished my bachelor's degree, came back to the US, and I did a master's in bioinformatics. And there, it's what I got at the time. So I worked with cattle research. So we're trying to make beef better, trying to narrow down some genetic regions that could be associated with better beef, more muscular animals with more marbling score and a lot of animal science terminology there. And then that's where I learned a lot. It started my basis for computational research and bioinformatics work. When I came into my master's, I didn't have any coding experience or anything like that. And then after that, I joined WashU, and that's where I went back to the oncology world. So I got my PhD here at WashU. I defended last year at Dr. Lee Ding's lab. And I plan on staying in oncology forever now. No more changing.
Yeah, that's impressive, Fernanda. And your journey is quite inspiring. Could
Thank you.
you could you explain your research in layman's terms?
So my research, which is what I did during my thesis work and what I continue doing now as a postdoc at Dr. Ding's lab, it's about predisposition to cancer. So basically saying we know a lot of what we do to ourselves that could lead to cancer, like smoking, exposure to radiation or things like that. They're environmental factors, but we know very little of what is inherited from our parents in terms of mutations and genetic events that could lead to a higher probability of developing cancer. So there's a lot of research on that. A lot is known for breast cancer and colorectal cancer, prostate cancer, which are very well-studied cancers, but other cancer types, we know very little. And research is needed on that so that we can detect it earlier and prevent it in a better way. So my research is studying things that are passed along through generations, so rare mutations that could be involved in predisposition to cancer. And some of my studies also involved families that had multiple myeloma, so sort of a segregation, generational analysis. So
Yeah.
very basic terms, that's what I do.
Yeah, thank you for breaking down so clearly. It sounds like your work has significant implications. Yeah. So, how does your work as a biometrician and computational biologist contribute to your understanding of cancer?
So I think, at the lab, we do have a 50-50 type of setup now where we do generate our own data and analyze our own data. So there's people doing wet lab and dry lab. In terms of the computational part, I think we can understand a lot of cancer development and progression by the analysis of genomic data. And the lab has a wide variety of data sets that go from genomic to transcriptomics, proteomics. Now we're moving into spatial transcriptomics and all of the single cell data sets that are evolving and becoming more and more intricate. So I think that really helps us understand how cancer starts, how it progresses, how it worsens, and so that we can better inform people and clinicians about what treatment routes they should choose or stay away from based on a more personalized information that is based on analysis of these people's genetic information.
It is fascinating to hear how your research is pushing the boundaries of our knowledge on cancer.
Thank you.
Could you tell us about the potential clinical applications of your research findings in cancer prevention or treatment?
So for my specific projects, I think it's mostly at the we work at the really detecting early context, right? So we want to inform people more about what they have that sets them up for a higher probability of developing certain types of cancer so that they can make informed decisions on how they want to change their lifestyle or what they want to look for and look for prevention strategies so that they can prevent developing it or catch it early to get a
Yeah,
better outcome. So I think there's a lot to be done, and the lab is, you know, fastly evolving through that. So now one of the main projects that I work for and that it's like most excite me on that is the first time that we're working on a project where we analyze the data and return those results to patients with the help of genetic counselors. So there's this consortia that's called PCGS. So it's called Patient Engagement and Cancer Genomic Sequencing, which is part of the Moonshot program
mhm,
by the government. And WashU is one of the sequencing centers. So we study three cancer types: myeloma, cholangiocarcinoma, and colorectal cancer. And we are focusing on underserved populations. So WashU is studying exclusively individuals of African ancestry. There's other centers, such as the center in Albuquerque, who only focuses on indigenous communities of New Mexico. And so we're really trying to get these people data sets, trying to investigate what they have at all genomic levels. But my part is looking at germline information, so predisposition, predisposing variation that could be specific to that population, and then hopefully we can return these results to them with the help of genetic counselors and say, well, maybe you should, watch for developing a breast cancer because you have this and this and that mutation. And so these people have it's a really like bench or I say computer to bedside
mhm.
type thing where we analyze the data and then these results are returned to patients, which is the first time that we're doing that. As a student, I didn't get the chance to do that. Now, as a postdoc, it's kind of interesting to get that. Also, getting feedback from genetic counselors of what we should improve in our analysis and how we should report those results better. So it's pretty cool setup.
Yeah, that's awesome. The application you mentioned could make a real difference in patient outcomes. And it's
Mhm.
exciting to hear about the practical impact of your work. How do you use computational approaches to analyze large scale genomic data in your research?
So as I said, the lab is we're really touching everything at this point. So we do work at not only like a data analysis, more bioinformatics focus context, but there's also a lot of tool developing in the lab for analyzing imaging data, for example, for spatial transcriptomics. That there are some tools out there, but they're not, the best. Or
Mhm,
there's always a caveat of everything that you're going to use. So in my context, we use a lot of internal computational tools for example, automating classification of variants. So a lot of germline information that we get. So we call mutations from individuals and we want to know what is a predisposing variant, right? And there's a set of guidelines that is published to do the classification, and that's usually done manually by clinicians or genetic counselors. And we can do that in an automated manner so we can analyze more variants and return those results in a faster way. And we're constantly improving those approaches to include more information from these patients that we can better classify them based on these guidelines. So that's just one example of using
mhm.
these computational approaches.
Yeah, I also believe that the use of computational tools in genomic is truly revolutionary. And your work is the perfect example of that. So, what role does machine learning or artificial intelligence play in your computational biology research?
So I think the field is just moving that direction, right? Everything is AI, everything is machine learning these days, and it's getting to be more and more. For my specific research, we are currently trying to use machine learning to better classify variants. So the tool that we have right now uses sort of a scoring system. So if it falls under guidelines A, B, or C, we apply a score, and then the sum of that score is what helps us identify if a variant is pathogenic or benign, right? And so that's basically based on the cohorts that we get. So we're trying to automate this tool and better develop this tool to become a machine learning algorithm where we can use publicly available data sets and, you know, data sets that are curated by genetic counselors and clinicians for classifying these variants so that we can use that as a training set to better classify the variants that we get from the patients from our internal cohort. So that's like one example. Machine learning is also being used in the lab for analysis of imaging data from these patients. So we're just currently evolving and developing more, but there's
Mhm.
a lot to be done.
Yeah, AI is a game changer in many fields, and it's great to see that it has application in your research. How do you see the field of cancer genomic evolving in the next 5 to 10 years, and what impact might it have on patient care?
I think there's a few things. One, I think the field is starting to finally focus on underserved populations. So most of what
Mm,
we know in terms of cancer genomics is based on European, white populations,
mm,
and that is not really applicable to individuals of African ancestry, to indigenous populations, to Asian populations. So I think the field is evolving to getting, a more diverse reference genome, for example, and that could help us better classify variants. And really focusing in recruiting these populations and making studies that are specific to these populations,
mm,
like what we're doing here at WashU. So I think that's like one step of like what the field is moving towards. But then also, I think machine learning and AI is playing a role into um, really, I think in terms of precision medicine is really going to help us better understand, even from, when we went to the conference, AACR conference in San Diego early in the year, everything was about AI and using AI to even scan through,
yeah.
literature and make the word of clinicians faster to, decide on treatment strategies, or use of AI to analyze X-rays or just images from uh, all sorts of different tests that are done on patients. So I think it's just going to be a combination of both, of like including a more diverse population and use of AI to better train these models and
Mhm,
hopefully understand more of how genomics applies
mhm.
beyond, what we know now, which is very limited.
Yeah, yeah, it will be interesting to see how these advancements unfolds. So let's talk about your personal life now. What do you do after work?
I think that changes over the years. Now I'm really into pickleball. It could change later. I used to do salsa a lot back in the day, and then I stopped that because, it's summer and you want to be outside and you want to um, play, but also just really relaxing and hanging out with my friends, like spending some quality time with my husband,
Mhm.
and doing, exploring St. Louis too, which, when you're a grad student, you don't really have time or money to do that. And now it's better. You can actually explore the city and do fun stuff. So, but yeah, I think currently I would say pickleball.
That is wonderful to hear about your hobbies and interests outside your work. And it is important to have a balance. Yeah.
Yeah, can't do
Based,
all science.
yeah. Based on your personal experiences, what advice would you give to our audience about life?
About life. I guess don't worry too much about tomorrow. I think I think scientists in general tend to, because we're trained that way, right? We're
Yeah,
trained to
we fail every
think
day.
about, yeah, like we just think about everything in very much detail. And sometimes I think a lot of my friends that are in the field overthink stuff a lot because we're
Mhm,
trained to do that and analyze things. And sometimes, like, I think we worry so much about like just like regular life things that don't need to worry too much about.
mhm,
So I think it's just, don't worry too much. For like a scientist, I would say trust the work that you're doing. It
mhm,
sometimes it feels like very insignificant and small, but at the end of the day, you are helping somebody out there who is suffering from cancer and just is hoping that somebody comes up with a cure or some better treatment or some better, um, you know, prevention strategy or whatever you want to do. So
mhm,
you're helping. It may seem like a very tiny little paper that you publish in a journal, but it's having an impact on somebody for sure. Even if it's not now, it's going to be later.
mhm.
Um, so
Yeah,
yeah, and have
yeah.
fun with it.
Yeah, thank you for sharing such valuable advice. I'm sure our audience will find it insightful.
I hope so.
Thank you, Fernanda, for sharing your insights and experiences with us today. It was a pleasure having you on the podcast.
Thank you for inviting me. It's good to talk about what we do in a more, relaxed setting and just share what we do and what we think about. So thank you for
Yeah,
inviting me and considering me.